So today what we are going to see is we are going to see about creating CDNA library. Okay, so from CDNA library we need a starting material as mRNA, isn't it? So mRNA which will be converting into DNA. So that DNA we are going to call it as CDNA. Understood? So basically when I create a CDNA, so what it is going to be now? So it is going to be a blunt end, that is mRNA is going to be blunt. So when I create a CDNA, it is also going to be a blunt end, isn't it? So it is very difficult to ligate into the vector, because if it is a sticky end, it is very easy, right? But if it is a blunt end, it is very difficult. So in order to convert the blunt end into sticky end, what people will use now, it is called restriction site linker or adapter, which makes more sticky. Understood? So mostly what they will use now, their adapter. So what they will use now, they will use a ligases which is from T4 DNA. So they will add into the end of the cDNA, and then what will happen now, this adapter will have a specific restriction site. So they will have a, so this is, if this is your blunt, one second guys, so this is your blunt end mRNA. So in that they will add a adapter to both the end. This adapter will have a restriction enzyme. So when they use T4 DNA ligase, so it will cut like this. They'll cut like this. Okay, so now by creating this sticky end, it is very easy to ligate into a vector. Understood or not? Guys, can you imagine what I'm saying? It is literally converting blunt end into a sticky end. So once the sticky end is produced, now it is very easy to ligate into a plasmid. Once a plasmid is ligated, the recombinant plasmid can be transformed into an E. coli. Okay, so now this is how they'll create a cDNA library. So how it can be done now, this is what exactly until now I'm talking. So this is your double stranded cDNA, which is blunt end. So they will add a linker or adapter which contains a restriction enzyme. So this restriction enzyme is a BAMH1. So when they ligate BAMH1, after that they use a specific restriction enzyme, they'll create a sticky end. Now this sticky end can be easily ligated into the plasmid. So this is called recombinant plasmid. Understood or not, guys? Here one more thing what you guys have to remember is this mRNA has a capacity to express. So this plasmid will be a expression plasmid. What extra it will have? It will have a promoter. Understood? So when it is been ligated, this protein will be, this mRNA will start synthesizing your protein. Understood? So the next question, how I am going to see what is the condition what they will do now. Let me give you a scenario now. I will take a 2 set of test tube. I want to know if this bacteria can grow at 60 degrees Celsius or not. Guys, can you imagine what I'm saying? Basically, bacteria will grow at 37 degrees Celsius, isn't it? So I am incubating one bacteria at 37, another one is 60. So after overnight, I am isolating all the mRNA, converting into cDNA. and then I created a CDNA library. So now I want to screen is there is any extra specific protein has been expressed when the bacteria is incubated at 60 degrees Celsius. Understood or not, guys? Are you following what I'm saying? So how I'm going to screen? There are two methods by which I can screen a genomic library or CDNA library. One is colony hybridization, other one is block hybridization. I want everyone to listen carefully because based on this there was so many questions asked. Okay. So now do you remember what is this plate? After transformation, the bacteria which is plated in a selection media which contains an antibiotic. So all these bacterial colonies are recombinant. Understood? That I am calling it as master plate. So now what I am going to do is I am going to take a nylon membrane, circular nylon membrane. Mostly the nylon membrane is positively charged, whereas cell surface bacteria is negatively charged. So carefully what I am doing is I am going to take this nylon membrane and then carefully I am going to place it on the plate. So by doing so, what happens now? One or two cells from each colony will be sticked. The position will be same. Understood or not? You guys are following what I'm saying? Yes, exactly. Impression of colony. So now after that, what I'm going to do is I'm going to treat this nylon membrane by cell lysis buffer. That is nothing but your lysozyme and then EDTA. By doing so, what happens now? All the cell will be lysed. Once the cell lysed, the DNA will be exposed. Now the DNA is positively charged, whereas the nylon membrane is negatively charged. So now all the plasmid DNA will be immobilized. Okay, so after that, so what we want to do is, after when the DNA is exposed, we know we are hypothesizing some DNA, some gene. So here what I hypothesize na beta globulin gene. So even though it is not related, for example, I am saying I am looking for a beta globulin gene, whether it is expressed, if it is expressed, which colony it contains. So what I will do now, I will take a piece of DNA which is complementary to the beta globulin gene. I will radio label it. After radio labeling, what happens now? I literally just pour this radioactive label on this nylon membrane and then allow it for some time. This probe will go and bind to the complementary strand, and then if I expose it to X-ray film, that is autoradiogram or chemiluminescence, I will get a spotlight. What it says now, the corresponding colony contains my gene of interest. Now I can take these two colonies from the master plate and then I can use it for my further studies. This is called colony hybridization. So Gayatri, your question is how to create double stranded single stranded. Na? So by addition of NaOH, all the hydrogen bond will be broken. Okay. So now double stranded will be converted to single stranded. Okay. So this is about colony hybridization. The next one, what we are going to discuss is plug hybridization. So plug means what? Can you guys, do you remember what is meant by plug? What is called plug formation? In the bacteriophage lysis, we are going to call that area as plaque formation. Very good. Bacteriophage lysis zone. So now what I am going to do now, very carefully, I am going to take this membrane and then keep it on this surface of this media. So now what will happen? Some of the bacteriophages will come and attach. Okay, and then one more thing here is, after attached, all the bacteriophages single stranded DNA. After lysis, all the single stranded negatively charged DNA will be lysed, and then it will be hybridized. So now what I can do now, I can add the radioactive label probe, and then now I can take the corresponding lysis, corresponding bacteriophage contains my gene of interest. Guys, understood or not? That is a colony, we are taking bacteria. Here it is a bacteriophage, we are taking, it is a lysis, we are taking a bacteriophage. Okay? Any questions, guys? If no questions, I can go on to the next slide. Okay. So how many of you performed? Process is same, Chitra, process is same, but Nitish, bacteriophage virus will have only single stranded DNA. There is nothing called haploid genome. So your question is haploid genome and then single stranded DNA same? It's not. Haploid genome, we will call it as if a cell contains only half of the chromosome. 23 pairs, if it is have only 23 chromosomes, then it is called haploid. Don't get confused. So those who are not performed southern blot, let me show you a picture.
Each contains plasmid DNA, but the plasmids in one isolate may not be the same as the plasmids in another isolate. We can purify the plasmid DNA from the isolates and use two procedures, gel electrophoresis and southern blotting, to compare these plasmids. As a first step, we can use gel electrophoresis to examine the plasmids in their native form. Blue loading buffer is added to an aliquot of the plasmid sample, and the mixture is loaded into wells of a jello-like slab made of a polysaccharide material called agarose. The agarose contains microscopic pores through which the DNA can travel. An electric current draws the negatively charged DNA through the gel toward the positive electrode. The positions of the DNA on the agarose gel indicate the sizes of the plasmids. Smaller pieces of DNA migrate faster than larger ones through the agarose pores, because larger pieces meet with more resistance. In this example, the plasmids in lanes A through D are similar in size. The plasmid in lane C migrates slower than the others and is therefore a little larger. Although the plasmids in A, B, and D appear to be the same, we must perform other tests before we can come to this conclusion. Our next step is to add enzymes called restriction enzymes to the plasmids. An example of a restriction enzyme is EcoR1. A restriction enzyme moves the DNA at a particular recognition sequence. EcoR1 recognizes and cuts the sequence GAATTC. If the plasmids in the different tubes are identical in sequence, they should have the same number of EcoR1 recognition sequences. The type of plasmid in this particular sample can be cut in 4 places with EcoR1, producing 4 fragments of different sizes, represented here as different colors. When the fragments are loaded on a gel and electrophoresed, the fragments separate from each other. The slower bands on the gel contain larger fragments, and the faster bands contain smaller fragments. The patterns of fragments in lanes A and B appear identical. On the other hand, the patterns of fragments in lanes C and D appear unique. Are the DNA sequences and fragments in lanes A and B the same or nearly so? And is there any similarity between them and any of the fragments in lanes C and D? To answer this question, we perform what is called a southern blot. An apparatus is set up to transfer the DNA in the gel onto a solid support, such as nitrocellulose paper. The gel is placed on top of a sponge, soaking in an alkaline solution. Nitrocellulose paper is placed on top of the gel, and a stack of paper towels is added on top of the nitrocellulose. The solution is drawn up the stack. The DNA denatures in the alkaline solution as it's carried up the stack. The DNA stops traveling when it reaches the nitrocellulose. If we could see the DNA on the nitrocellulose paper, it would show the same pattern of DNA bands as existed on the agarose gel. We place the nitrocellulose paper into a plastic bag and add a solution containing a radioactive single-stranded DNA called a probe. In this case, the probe consists of all the fragments from plasmid A. During an incubation period, the probe anneals to complementary sequences, forming base pairs with the DNA in some of the bands on the paper. The excess probe is washed off. The only probe that remains on the nitrocellulose paper is the probe that has annealed to complementary DNA on the paper. After the probe is washed off, the paper is overlaid with X-ray film. The film and nitrocellulose are kept in the dark for a period of time, and then the researcher develops the film. The radioactivity in the probe creates black bands on the X-ray film, which is called an autoradiograph. The autoradiograph provides more evidence that plasmids A and B are the same or very similar. However, plasmid D is unrelated to plasmid A, from which the radioactive probe was derived. Plasmid C has some similarity to plasmid A, but the similar sequences are found on different sized DNA fragments. The autoradiograph gives us more information than the agarose gel on its own.
Guys, clear? Got an idea what I'm talking about? Let me see if I find any. Very good, guys. We're just looking for DNA library construction, animation, anything is there? Just 1 second. Southern hybridization. The word hybridization means binding of probe with DNA. Southern blotting means the technique is called southern blotting. Okay, both are same. It happens in the okay. Let me, I got an animation for DNA library. Okay, can you see the slide now?
How do researchers study the vast amount of DNA in the cells of an organism?
Guys, you listen carefully. This video will show you will give you the situation and then it will explain. It will be very nice. Please enlarge. This is the only thing, Mamita. I cannot enlarge. Okay.
How do researchers study the vast amount of DNA in the cells of an organism such as a human? One way is to create a DNA library, which is a collection of DNA fragments that come from one organism and that are stored in another organism, often a bacterium. To create a human DNA library, a researcher begins by extracting and purifying DNA from human cells. Purified DNA consists of extremely long DNA strands. To begin working with the DNA, the strands must first be cut into manageable sizes. The DNA is therefore digested with restriction enzymes, which cut the DNA at specific sequences. The restriction enzymes cut the DNA into thousands of smaller fragments, each of which may contain one or more genes. Although we show just a few fragments, let's suppose they represent all of the DNA that exists in human cells. The colors indicate that the fragments are different and have unique DNA sequences. To create the library, each fragment must be inserted into loops of DNA called plasmids. A plasmid is a type of vector that allows the DNA to be shuttled into bacteria. The plasmids are digested with the restriction enzymes and then sealed to the human DNA using the DNA ligase enzyme. The resulting molecules are recombinant. The recombinant DNA molecules are added to bacteria, and the bacteria are made to take up the DNA. When the bacteria have taken up the DNA, the entire collection of cells and DNA represents a human DNA library. The recombinant plasmids remain separate from the bacteria's own DNA, making it easy to extract again. To analyze any of the DNA fragments, the bacteria are grown on a rich medium that allows them to reproduce and make more of the recombinant DNA. This form of the library allows a researcher to analyze unique human DNA fragments and locate the fragments that contain the genes that the researcher wants to study.
Shall I repeat one more time? Okay. This is today, it is managed, it is a lot of people.
How do researchers study the vast amount of DNA in the cells of an organism such as a human? One way is to create a DNA library, which is a collection of DNA fragments that come from one organism and that are stored in another organism, often a bacterium. To create a human DNA library, a researcher begins by extracting and purifying DNA from human cells. The purified DNA consists of extremely long DNA strands. To begin working with the DNA, the strands must first be cut into manageable sizes. The DNA is therefore digested with restriction enzymes, which cut the DNA at specific sequences. The restriction enzymes cut the DNA into thousands of smaller fragments, each of which may contain one or more genes. Although we show just a few fragments, let's suppose they represent all of the DNA that exists in human cells. The colors indicate that the fragments are different and have unique DNA sequences. To create the library, each fragment must be inserted into loops of DNA called plasmids. A plasmid is a type of vector that allows the DNA to be shuttled into bacteria. The plasmids are digested with the restriction enzymes and then sealed to the human DNA using the DNA ligase enzyme. The resulting molecules are recombinant. The recombinant DNA molecules are added to bacteria, and the bacteria are made to take up the DNA. When the bacteria have taken up the DNA, the entire collection of cells and DNA represents a human DNA library. The recombinant plasmids remain separate from the bacteria's own DNA, making it easy to extract again. To analyze any of the DNA fragments, the bacteria are grown on a rich medium that allows them to reproduce and make more of the recombinant DNA. This form of the library allows a researcher to analyze unique human DNA fragments and locate the fragments that contain the genes that the researcher wants to study.
Okay, hope it was clear, guys. Questions? Guys, shall I do that one more? CDNA library, if you are interested. I'm not getting response from everyone. Hope everybody started sleeping.
In most eukaryotes, the expressed segments of the gene, called exons, are separated by intervening sequences of nucleotides called introns. The exons and introns are transcribed by RNA polymerase, generating what is called precursor messenger RNA. The introns are then excised from the precursor messenger RNA, and the exons linked together to form the mature messenger RNA. Prokaryotic cell DNA does not contain introns, and prokaryotic cells are therefore unable to remove the introns from eukaryotic DNA and make functional messenger RNA. Therefore, eukaryotic DNA cannot be cloned directly into prokaryotic cells to make useful eukaryotic proteins. Before eukaryotic DNA can be cloned into prokaryotic cells, the mature eukaryotic messenger RNA must be isolated and used to make DNA without introns. The enzyme reverse transcriptase is used to convert the purified eukaryotic messenger RNA into double-stranded DNA. The resulting DNA, called complementary DNA or CDNA, can then be cloned into a bacterial cell. Genes cloned as CDNA can be transcribed and translated by the bacterial cell machinery.
Okay, this is about CDNA. Hope it was clear, guys. How introns are removed? Introns will not be removed because introns will never express. Chitra. Want to see replay CDNA? 5. How many of you want? How is mRNA isolated? Chitra, Gayatri, did you attend my DNA? Attend DNA synthesis, guys.
In most eukaryotes, the expressed segments of the gene, called exons, are separated by intervening sequences of nucleotides called introns. The exons and introns are transcribed by RNA polymerase, generating what is called precursor messenger RNA.
Sorry, guys.
The introns are then excised from the precursor messenger RNA, and the exons linked together to form the mature messenger RNA. Prokaryotic cell DNA does not contain
Okay, guys, can you see the slide now? Yes, I'll replay
In
one more.
most eukaryotes, the expressed segments of the gene, called exons, are separated by intervening sequences of nucleotides called introns. The exons and introns are transcribed by RNA polymerase, generating what is called precursor messenger RNA. The introns are then excised from the precursor messenger RNA, and the exons linked together to form the mature messenger RNA. Prokaryotic cell DNA does not contain introns, and prokaryotic cells are therefore unable to remove the introns from eukaryotic DNA and make functional messenger RNA. Therefore, eukaryotic DNA cannot be cloned directly into prokaryotic cells to make useful eukaryotic proteins. Before eukaryotic DNA can be cloned into prokaryotic cells, the mature eukaryotic messenger RNA must be isolated and used to make DNA without introns. The enzyme reverse transcriptase is used to convert the purified eukaryotic messenger RNA into double-stranded DNA. The resulting DNA, called complementary DNA or CDNA, can then be cloned into a bacterial cell. Genes cloned as CDNA can be transcribed and translated by the bacterial cell machinery.
Okay. Yes, exactly. Okay. Okay, guys. At what temperature southern blotting is done? It is done at different temperature. Renaturation, denaturation, and hybridization will be at 65 degrees Celsius. Jyoti. Shall I move on to the next slide? Constructing CDNA library. The same thing we have discussed. So this is your mRNA, which has a poly A tail. The mRNA is converted to CDNA. Now this is the CDNA. Now the CDNA will be having the linker, and the linker will be added sticky end. Now sticky end into lambda CDNA cloning. So this is what till now we have discussed about how to create a CDNA library. Just go through the slide for 1 minute. If you have any question, let me know. All the points we have discussed in detail. Once you are done, let me know. I'll go to the next slide. That is a method, Abhishek, so that the mRNA region will be intact. If you see that, it will be TGG, and then they will add the sequence so that it will be intact. If linker is not there in CDNA, it cannot be cloned into the vector. Sticky end na restriction site. It's not like that, Anushka, because as I told you, RNA is more unstable when compared to DNA. So when I add NaOH, it will be degraded very fast.